So thank you very much for staying after lunch. I know it's difficult at this time. I changed a little bit the talk because originally I was supposed to speak about role of structural plasticity in chaperones, but I will speak about it, but I will also tell you another story which actually combines stress response and protein homeostasis during aging, and this is a work that we do together with Tom Rabbit. So in my lab, we are trying to understand how do cells cope with different types of stresses. It can be either environmental changes or changes due to pathogen invasion or just life, the changes levels in pH, oxidants, salts, etc. And all these conditions can lead to protein misfolding, as we heard before. One of the mechanisms of cells to deal with protein misfolding is molecular chaperones. We spoke about degradation, but now we will speak about how to save proteins. So during non-stressful conditions, majority of proteins folded. Some of them have their own turnover, like we heard before. But during specific stress conditions, some of the proteins became misfolded, and this misfolding can lead to aggregation, which is very toxic to cells. So molecular chaperones can prevent this aggregation by recognizing misfolded intermediates and making stable complexes during stressful conditions. Upon return to non-stressful conditions, some of the proteins can be refolded in ATP-dependent manner, and some of them will be degraded. Historically, we can classify chaperones to many different classes, but we specifically like in my lab two specific definitions, and they depend on ATP dependence of chaperones. So one class of chaperones, which majority of you know, and it's widely explained in textbooks, ATP-dependent chaperones, such as DNAKJ, HSP 70, 90, 100, etc. So these are usually big complexes that need to cooperate with cofactors and co-chaperones, and they depend on ATP hydrolysis. This is one of such examples, HSP 70, or DNAK in bacteria, and you can see that it's a very complex protein and has two different domains, one that binds ATP and one that binds substrate. And these two domains need to change and have conformational changes in order to bind substrate and release them. In case of DNAK, DNAK needs to cooperate with DNAJ, which actually recognizes the substrate and brings it to DNAK. This is the structure of DNAK, and again, you can see there's two different forms, high affinity state and low affinity state. And definition of the state for the substrate is defined by ATP incorporation. The conformational changes that you can see here in the state, in high affinity state, the complex is really not folded, but it's very wide when it became very compact in low affinity state. And the changes is due to the small region of the linking region that brings these two domains together. Another type of chaperones, which we especially like in my lab, the ATP-independent chaperones. They're completely different. They're usually small chaperones, low molecular weight. It can be 70, 30, 40 kilodalton proteins. They do not have ATP hydrolysis facility. Many cases, they can bind substrate with very high affinity, but they cannot release them. So they're kind of holding chaperones. In many cases, they do not have any co-chaperones, but eventually they will need to deal and transfer the substrate to foldases or ATP-dependent chaperones. These are two examples of such ATP-independent chaperones. One of them is highly known and very abundant in many different organisms and tissues. This is small Hitchcock proteins. There's many different types of small Hitchcock proteins. All of them have this crystalline domain with intrinsically disordered regions, which are important for substrate recognition and for oligomerization of the chaperones. Another chaperone that I worked quite a lot during last 5-7 years is HSP33 chaperone. It's folded in the inactive form and became rapidly unfolded upon oxidative stress conditions. So the holdases and foldases need to cooperate together in order to prevent aggregation of proteins during stressful conditions. And we can show it in a following scheme when after specific stress, some of the proteins became damaged, the first line of defense. This is the co-chaperones or whole daisies that can make very stable complexes with a wide range of substrate. In cases of whole daisies, and this is ATP-independent process, and the complexes with whole daisies, they are usually very stable. We can keep them for a couple days in the lab, and there's no aggregation of this misfolded protein. The second layer is the fold daisies, which accepts misfolded proteins from whole daisies, and they can refold them in ATP-dependent manner or degrade them. And there's many questions in this scheme. First of all, what is the specificity of whole daisies, how they can transfer substrates, what whole daisies can be specific to stress or cannot be, if there is any specificity to stress response. And some of these questions we would like to address, and I will show some of the results. ATP-independent chaperones are quite diverse. There's many different modes of activity. Some of them are highly conserved, some of them found only in specific type of organisms. And today we understand more and more about the activity and the function. So there are several examples of ATP-independent chaperones and the conformational changes between inactive and active form. There's examples of intrinsically disordered proteins, LIA proteins. They are always unfolded in the active and active and inactive form. They're found in seeds and important for drying conditions of the key proteins like BSA, but unfolded. There's another type of chaperones which usually found in periplasm. They're actually folded and they have this hydrophobic cleft that can bind a wide range of substrates, but in periplasm where there is no ATP. So the ATP-independent chaperones that can also release substrates very slowly and refold them. Another type of activity is for Hitchcock proteins or gas-free chaperones. They're usually monomers or dimers in an active form, and upon stress response, they form high oligomeric structures. And usually it's done by this intrinsically disordered region that they have on C-terminal or N-terminal domain. So there is conformational changes or mainly oligomeric changes upon stress response. And another type of chaperones which we are working in the lab is this conditionally disordered chaperones, which are inactive and folded in during normal conditions and became rapidly unfolded upon specific stress conditions. And there are several examples of such proteins, many of them which we know today are bacterial chaperones. And I will show you briefly how we can actually exchange between these two modes and make HSP33 chaperones that have these oligomeric structures, and does it good, what does it do for activity of HSP33. So this is two examples of conditionally unfolded chaperones, HSP33, and another one is HDA. This is periplasmic chaperone, which is induced by acidic stress. During normal pH, it's very folded, has dimers, very folded and very stable, but at low pH, it became monomer and highly disordered. And this is actually the active form of HDA. But today we'll focus on HSP33. It's inactive in the reduced form and compactly folded and became unfolded upon oxidative conditions. In the lab, we can easily monitor chaperone activity. Yes, of course. What? Okay. It's on. It's green. Okay, I will try to speak louder. In the lab, we can monitor chaperone activity quite easily by monitoring aggregation by light scattering. And this is aggregation of citrin synthase, which is one of the unstable proteins that we're working on. When we add oxidized form of HSP33, you can see that there is no aggregation because it binds to misfolded intermediates of citrin synthase and prevents this aggregation. What was interesting is that if we add very small amount of DDT, we abolish this activity of HSP33. And it was interesting because HSP33 is reducing chaperone. It's a cytosolic protein, and cytosol is reducing environment. So how come reduction of cytosolic protein leads to its inactivation? And apparently it's physiologically relevant because HSP33 found in many different bacterial and pathogens, and all of them need to deal with very low concentration of oxidants such as peroxide, hypochloric acid, which is also found in bleach, and all of them activate HSP33. The Jacob lab where I did my postdoc showed very nicely that knockout of HSP33 in E. coli and also in Vibrio leads to decrease in survival of bacteria in very low concentrations of oxidants. We also did bioinformatic analysis, and we showed that there is more than 2,000 homologues in bacteria of HSP33, some of them in aerobic and anaerobic species. And there's also few homologues in eukaryotes, which are shown here by stars. What was interesting that Archaea doesn't have any of HSP33 homologues, and we were kind of puzzled why. And one of the reasons that we think is that Archaea doesn't have very conserved DNA chaperone. And the reason that it might explain that HSP33 needs to transfer its substrate to the NKJ system, and if it's not really highly conserved in Archaea, so maybe it can explain why we don't see homologues of HSP33, because it doesn't have its partner where it can transfer the substrate. When we focused into eukaryotic homologues of HSP33, we actually found that many of them are unicellular organisms, and many of them either pathogens or organisms that need to deal with extreme environmental changes. And we were kind of puzzled with this specific group of pathogens, Trypanosoma and Leishmania. Many of HSP33 homologues are found in this family. There's 15 different homologues, and they found in Trypanosoma and Leishmania. And Leishmania, some of Israelis at least know from here. Leishmaniasis, Shoshanati Yerecho, and Trypanosoma also leads to many different infectious diseases, and none of them actually have cure today. So we wanted to understand why we need HSP33 and why it's so conserved in this family. And one of the reasons that we thought that actually all these pathogens need to have two different environments. They live in insects and in bloodstream. And transfer between insect to bloodstream is accompanied with changes in environment, in oxidative stress and heat response. So in collaboration with Shula Michaeli and Rosie, which is sitting here, she established a Trypanosoma system in my lab, and we did silencing of HSP33 homologue in Trypanosoma, and we see that silencing of HSP33 leads to decrease in survival of Trypanosoma in very low concentration of peroxide. But majority of the data that we have today is from bacterial HSP33, and some of the structural experiments I will show you. So what we have a structure of inactive form of HSP33 from Bacillus, and we did also model of the same structure on E. coli. We can define 3 different domains in HSP33 based on this structure and also functional studies. There is N-terminal domain, which is very folded, very hydrophobical domain, and the C-terminal domain, which serves as a redox sensor. It has 4 completely conserved cysteines. In all these 2,000 homologues that I showed you before, all of them have these 4 completely conserved cysteines. At least in bacteria, we know that they bind zinc in the reduced form, and this high affinity between zinc and cysteines keeps this protein folded and inactive. There's also this middle domain that for many years was named as linker. But today we think that it actually has additional function in inactivation of HSP33 and also sensing thermal conditions, not only oxidation. We also did predictions of disordered regions on this chaperone, and you can see that the C-terminal domain have high propensity of intrinsically disordered region features, and the middle domain is kind of partially unfolded. So how does HSP33 works? Upon oxidation, zinc is released, and now 2 disulfide bonds are formed. It leads to completely unfolding of C-terminal domain. We have NMR structures of the C-terminal domain, and we see that it's folded in the reduced form and became highly unfolded upon oxidation. The disulfide bond formation leads to destabilization of middle domain, and this is the active form of HSP33 that can bind a wide range of substrate and prevent the aggregation. And these complexes are quite stable. We leave them for a couple of days in the lab with no aggregation of the misfolded proteins. Upon a reducing condition, when we just add small amount of DDT, the C-terminal is refolded, and this refolding leads to destabilization of the substrate, and transfer it to the folding system, to the DNA KHA system for further refolding. And during last years, we asked ourselves why we need this refolding, why we need this plasticity, why we need the unfolding of the substrate, what is the role of the structural plasticity in substrate recognition, in transfer, and release of the substrate. And we quite were puzzled. First of all, we didn't really know where these unfolded regions are, because we could not do NMR on this chaperone, and crystallography is also quite difficult for this. So we looked for mass spectrometry-based methods that can help us to map conformational changes on the chaperone and see what is why do we need them in activation and substrate binding. So one of the methods that we found very useful for this kind of analysis is hydrogen deuterium exchange when we do a following experiment. So we incubate protein in deuterium for different time points. It can be from 10 seconds to a couple hours, and the short time we have only few deuterium incorporated, and larger times more deuterium is incorporated. Then we can quench the reaction, do proteolysis, and look on specific peptides on the protein and calculate mass differences. So if this region is folded, it will not have deuterium incorporated in short time, only in the long time, but the exposed regions will have deuterium incorporated already in 10 seconds. So then in this way we can say what are the regions that are more exposed or less exposed or became more flexible. But we wanted to have actually better assignment and to get some idea about stability, not only on the exposed regions, but also to have some information about stability of the regions. And we developed a method together with Michael Filcheret in the Duke University, which is called SUPREX. So we do exactly the same experiment. We do a hydrogen deuterium exchange, but now in presence of two naturans, for example, guanidinium chloride. We do different concentrations from 0 to 5 molar. We usually use 12 different concentrations, and in each of them we did hydrogen deuterium exchange. So now we have differences in mass that correlates to different peptides within the protein, and we can make SUPREX curves, so stability curve, where we plot differences in masses, which means how many deuterium were incorporated over concentration of guanidinium. So there's kind of stability curve, which is shown here, that can be calculated for different regions over the protein, and here we can calculate C-half value that can tell us about stability of that specific region. From this we can also calculate free energy and evaluate stability of proteins. How does it can help us? We can compare two different folding states, or we can compare changes upon binding or upon post-translational modification. So if we compare two different states, we have comparison of two different SUPREX curves, and you can see that if this specific region is folded, it will have lower C-half values, and also the initial incorporation will be different. So at first we wanted to understand what happens to HSP33 upon oxidation. We knew from CD and from other studies that we have big changes in and unfolding of the protein, but we didn't really know where. So a combination of many different SUPREX curves I have shown here. At that point we had only N-terminal and middle domain coverage without C-terminals, and you can see that the most destabilized regions upon oxidation, they are this middle domain and part of the N-terminal domain. So many regions on HSP33 became destabilized only because of the disulfide bond formation on C-terminals. This is a SUPREX curve which corresponds to this part on the middle domain, and you can see that it's folded in the reduced form and became quite unfolded in the oxidized form. We did also several other analyses together with limit proteolysis, and now we have quite good coverage of HSP33, and we see that many parts of the N-terminal domain, middle domain, of course C-terminal domain, became unfolded only because of the disulfide bond formation upon oxidation. What was interesting, then we looked on the complex. We wanted to see what happens to the chaperone upon substrate binding. And then we saw that when we analyze the complex, we gain stability back on this region of the middle domain and partially on the N-terminal domain. So at that point, we thought maybe the middle domain is an interface for misfolded proteins, and this is our binding site, or maybe there's some refolding processes go up on substrate binding. Then we did many different analyses, and we mapped binding regions of HSP33 and looked on substrate specificity. In some of the data I can show you here, where we used in vivo and in vitro cross-linking, and we mapped amino acids which are important for substrate binding. And you can see that many of them actually can bind substrates, and they're very not defined. The interface of HSP33 with misfolded intermediates is quite broad. What does it bind? And it seems like HSP33 binds partially folded substrates. It doesn't bind intrinsically disordered proteins, and it also doesn't bind folded proteins. It needs some partial unfolding. And it was actually interesting because when we looked on the substrates by HT analysis, we saw that refolding of HSP33 leads to unfolding of the substrate and transfer to the DNA KHA system, which prefers unfolded proteins. So the transfer here occurs only due to changes in the structure of the substrate and not because of specific sequence of specific binding motif. So later on, we were also puzzling about inactivation of HSP33, because there is always quite high level of HSP33 in cells during normal and oxidative stress conditions. It's about 3-5 micromolar. And we know that we don't want to have always active HSP33 because then it will bind many partially unfolded proteins within the cell. And is it only the C-terminal domain that keeps the protein inactivated, or maybe there's additional domains that are important for this? And in order to answer this, the bright group of 3 master students, Ohad, Uded, and Moore, which Moore only here, looked on these specific interactions of N-terminal in middle domain. And they wanted to understand if this middle domain is important for inactivation of HSP33. So what they did is following, they replaced middle domain by many different sequences. One of the sequences has a reversed order. It has exactly the same amino acids, but in reversed order. So we keep amino acid composition, but the structure is different. Then we also did different chimera proteins where the middle domain was replaced by fully disordered region or by folded regions, and now we have many other chimera proteins, and they all behave quite similarly. So we looked on redox regulated activation, on anti-aggregation activity, on transfer to the DNA KHA system, and we found that, first of all, quite interestingly, contrary to the wild type, which is active only on the oxidized form, all our chimera proteins are constitutively active. So if you replace the middle domain, if you even do small mutation in that domain, it destabilizes connectivity with N-terminal domain, and we have constitutively active chaperone, which binds substrates quite efficiently and prevent aggregation of many different substrates. Is it good for the chaperone? What is it can transfer them further? Apparently not really. So what we can do, in this field, we're quite lucky, we can work with enzymes. And when they're unfolded, they don't have enzymatic activity. But if the enzyme is transferred to the DNA KHA system, we can monitor enzymatic activity with time. So when luciferase is unfolded and bound to HSP33, we don't have any enzymatic activity. If we add the DNA KHA system together with DDT, we can get some refolding activity. After 2 hours, about 60%. And all our chimera chaperones, all of them could not transfer substrates very efficiently. We get between 25 to 35 efficiency. So they kind of super hold this. They can prevent aggregation, but they cannot release quite efficiently. So next we wanted to understand why. And we did several analyses, and one of them is SEC mouse, where we can analyze native forms of chaperone and define the size by using light scattering and a reflective index. So in the wild type HSP33, in the reduced form, they're usually monomers in 33 kilodalton. In the oxidized form, we have small fraction of dimers and a little bit of high oligomers, but all our chimeras proteins, they have monomers and high oligomers in the reduced form, and in the oxidized form, they form many different high oligomeric structures which are very dynamic. There's many different oligomeric structures in this form, and it's quite not uniform. So we think that maybe these oligomeric structures, there is a reason for preventing substrate transfer to the DNAK system. So we think that HSP33 is kind of gatekeeper of HSP33, and perturbation of this domain leads to constitutive activity of HSP33 and to oligomerization. So our scheme today for HSP33 working cycle is following. During reducing conditions, the C-terminus is quite locked due to zinc and reducing cysteines, and the middle domain is folded quite nicely on top of the N-terminal domain. Upon oxidation, the C-terminal domain is unfolded, which leads to partial unfolding of the middle domain and to active form of the chaperone that can bind many different substrates. Upon reduction, the C-terminal is refolded, the middle domain is going back on top of the N-terminus domain, and the substrate is transferred to the DNAKJ system. If we perturbate the middle domain and we replace it by any sequence, we immediately get these high oligomeric structures, which are quite efficient as anti-aggregases, but they cannot transfer substrate very efficiently. So next, Ohad Yogev asked what actually happens in cell. What is the substrates of HSP33 in cell? And he looked on different species of HSP33 knockouts with plasmids of different HSP variants, did stress response, looked on soluble fractions, did IP, and analyzed interactome of HSP33 during normal and stressful conditions. So we found that there is approximately 100 proteins that bound to HSP33 to the wild type form, and there is difference between interactome and normal and stress conditions, which was already interesting that HSP33 can bind some proteins during normal conditions, and of course its interactome became higher upon oxidative stress conditions. The chimeric proteins, which have either disordered or folded regions, they also bound several proteins, and there was no big change between stress and normal conditions, which actually makes sense because we think that they're always active during normal and stressful conditions. When we actually looked on Z proteins, we saw that there is a subset of proteins which are bound only to wild type HSP33, which means that this middle domain is important for the activity. And when we looked on these proteins, they're usually stress-regulated proteins. They do not have any function enrichment, and we think that there may be in vitro substrates of HSP33. On the other hand, there is a subset of proteins which are always bound to HSP33 depending on the middle domain. And these proteins, when we look on the functional clustering, we see that many of them relates to protein synthesis or to redox homeostasis, and we think that maybe HSP33 has additional role that wasn't addressed up to now during normal conditions, and it will be actually very interesting to see why all these different chimeras of HSP33 recognize proteins which are shown here, and it seems that these interactions is middle domain independent. But HSP33 is only tip of the iceberg. It's only one of redox-regulated proteins which we know today that uses post-translational modification of its cysteines for the function. And we would like to see the whole picture. We would like to define other redox-regulated proteins and see what is their connectivity and what's their role in different physiological conditions. And one of them we are especially interested now, it's aging, and we want to understand why we get rusty with the age. On the other hand, why we became more and more oxidized. And is there any role of oxidation in protein homeostasis during aging? In order to answer on this question, Mital and Rosie and Ohad, the former postdoc in my lab, started a new collaboration with Tommy Rabid, and we are working in yeast in this project. But at first we wanted actually to monitor redox status. We know that cells became more and more oxidized with age, but we wanted really to quantify and to see what is the differences in population of yeast during aging. So we looked for a good in vivo sensor, and we were quite lucky to have very good collaboration with Tobias Dick and his former postdoc, Bruce. And they're working with different series of GFP sensors. So all those GFP sensors have this specific pair of cysteines that became oxidized upon oxidative conditions. And in addition, in this sensor, it fused to glutaredoxin. So if we have high level of oxidized glutaredoxin, it will oxidize glutaredoxin, and then it will be oxidation of raw GFP, and then we will get different spectrum in fluorescence. So this is the fluorescence of raw GFP when it's fully reduced, and this one is when it's fully oxidized. And you can see that there is two wavelengths which have completely different ratios for oxidized and reduced form of raw GFP. So the ratio between them can tell us what is the oxidative status of cells that expressing this raw GFP protein. So what we can do with this, we can go for microscopy or for plate reader, but we found that we actually use flow cytometry to quantify redox status of cells, of each cell independently, and also to sort cells based on the redox status. So we start with yeast culture and monitor two different wavelengths, 480 and 410, and the cells go through the laser. Then we can use detector and define for each cell what is the ratio of two different wavelengths, and define it if it's reduced and oxidized by definition, which we already defined empirically before it. So then we can ask the FACS to sort cells based on the redox status, and in this form we can have subpopulation of specific each culture that has groups of reduced, oxidized cells, or others that either don't have GFP or have different properties. So at first we wanted to see if we can use this method actually to quantify redox status of cells, and we used just wild type cells and monitored oxidation level during first 7 days. And you can see, as expected, with age cells became more and more oxidized. If we use mutant in glutaredoxin reductase, you can see that these cells already became oxidized from the beginning and more oxidized with time. But the fact what it gives us division to subpopulation. So we can actually to see distribution of cells and see if there are any subpopulation of oxidized and reduced cells during aging. So when we look on cells only in log phase, we can see that majority of cells are reduced with age of the first, second, third day. We can see that there are subpopulations of the reduced and oxidized cells. And at later point, we can see that actually there is two types of population. It's not continuous one. And we can now sort them based on the redox status for each day, for day 2, 3, and further, and have subpopulation of reduced and oxidized cells of the same age and look on their properties in growth cell, in proteome cell division, and further. And in this way, we want to understand what is actually more important here, age or oxidative status of cells, in order to define the properties. So to summarize the data, we did a lot of growth curves, proteomic analysis, etc. And we see that majority of the cells, as expected, are reduced. The population changes with the age. When we look specifically on the cells, we see that the reduced cells grow much faster. They have much lower doubling time than the oxidized cells. What was interesting is that many of the reduced cells are in the process of the budding, and we looked also on divisions, and we saw that the reduced cells, they have either they didn't divide or divided only once, while the oxidized, they usually divided more than one time. But what was interesting is that all these properties remain the same in the different age. So what was important here is not age of the culture, but the redox status of them. We did also proteomic analysis of the reduced and oxidized subpopulations, and we found that again they had exactly the same profile in different ages. And in all of them, we saw that there is a subset of proteins which are upregulated in the reduced cells and downregulated in the oxidized cells. And when we look specifically on the function, we see that in the reduced cells, many of the antioxidant proteins are upregulated, and the cells have more transcription, they are more alive. And the fact that they have many antioxidant cell proteins maybe suggests that that's why they became reduced and didn't transfer to be oxidized. We looked also on functions of the significantly different proteins which are abundant in reduced and oxidized cells, And we can see that many of them relate either to ribosome or to stress response. Some of them appear more when they're oxidized, or some of them when they're reduced. In addition, what we can do, we can mobilize cells and look specifically on each cell that expressing raw GFP and monitor oxidation with time during first 24 hours. And I would like to show you some movies where we can see that some of the cells, when the colors, when it's blue, then it's reduced, when it's red, it's oxidized. And you can see that with the time from the beginning, the cells, majority of them are reduced. They divide, and the daughter cell became also reduced. And further, we can also monitor the oxidative status, and we see that there's kind of threshold. So the cells can maintain the redox status up on 60–70% of full oxidation, but once they reach the threshold of 70 or 60%, so there is no way back. They became more and more oxidized. What happens to the daughter cells of the oxidized cell? And we saw that in many cases, when oxidized cell is dividing, the daughter cell will be also oxidized for quite long time. And this is one of the examples. These two specific cells, you can see them here. They're quite oxidized. I think the oxidity level here is approximately FS06, 60%. And the daughter cells that are formed, they're also oxidized exactly the same as the mother cells. So to conclude this part, we think that the yeast culture of the same age is heterogeneous. And we use facts to sort cells based on the redox status and define properties and features of these subpopulations. There's kind of threshold for oxidation for the cells, about 60–70%. Once the cell passes threshold, they cannot be reduced back. And we think that oxidation relates to division and to budding of the oxidized cells. And in the last part, really a couple slides, I would like to show you another story which is quite related to this one. We wanted to look on protein oxidation. We know that the cells became more and more oxidized, but what actually happens to the protein? What proteins became oxidized with age? And this is a nice study that started in Jacob lab by Nico Brandes, where he looked on chronological aging of yeast, and he took samples of first 5–7 days during chronological aging, and he looked on oxidative status of specific proteins. So we can quantify redox status of proteins, and for this we need a method that will be sensitive to cysteines and can distinguish between reduced and oxidized cysteines. It can be quantitative. And this method is called OxyCat. It uses two different isotopes, light and heavy ICATs, which are shown here. All of them binds to reduced cysteines by this reactive group, and they also have built-in as affinity tag to purify labeled peptides. The only difference between these two ICATs and this linker of carbons, which gives difference of 9 Dalton. So what we do, we start with proton, with light proton, and we incubate it with light ICAT, which binds to reduced cysteines. Then after reduction of the LICAT, of the sample, the oxidized cysteines that were oxidized in the original sample became reduced, and now we can trap them with a heavy ICAT. Then we do trypsin digestion, a mass analysis, and we're looking for two peptides that have exactly the same sequence, but they differ in 9 Dalton. And in this way, we can say what is the oxidative status of that specific cysteine and how it changed in different physiological conditions. So we did this analysis for chronological aging, and the results are shown here. Each line is different peptide, and oxidative status of the peptide is colored from blue to red. And you can see that during first 2 days, majority of the proteins are reduced, and there is collapse in oxidative status after day 3 or 4, where majority of the proteins became oxidized. This collapse can be postponed by either caloric restriction or by different mutations, where we always see the subset of proteins which are shown here, which became oxidized day before the collapse. And we thought maybe this protein somehow relates to the collapse, and maybe they are regulators of aging. And when we look closely on these proteins, we found that many of them relates to protein homeostasis, protein translation, and one of them is I is CDC48, which was discussed quite nicely by Tom yesterday. It has homologue in mammalian cells, VCP OP97, and it's one of the major regulators of protein degradation. As it was stressed yesterday, CDC48 is powerful machinery that pulls proteins, misfolded proteins from many different organelles and brings it to proteasome. So what we were puzzled, we said, okay, we have the specific systems which we know that they became oxidized day before the collapse. Let's mutate them to serine and see if they affect redox status of cells. And apparently what we found that while the wild type cells became more and more oxidized with age, our mutant cells remained reduced over 8 days and even longer. So just one mutation of specific tile in CDC48 protein leads to consecutive reduced status of cells over age. Is it good for cells if they became healthy and long-lived? Apparently not. They have high UPR response, and they're highly sensitive to oxidative stress conditions. They have slight decrease in longevity, and we also think that the specific mutation changed completely the interactome of CDC48 and its ability to interact with proteins. So now it's kind of, we would like to speculate, at least at this point, that this early age oxidation is beneficial for longevity of cells, and it will be actually very interesting to understand what is the mechanism. So to sum up, the first part of the talk, I spoke about structural changes in HSP33, which are important for its activity and substrate release. We think that there is several very interesting mammalian homologues of HSP33 that could be studied more closely. We design kind of super HSP33 by just mutating the middle domain, where we can get high binding of misfolded proteins but low decrease, and we think that this domain is important for inactivation of HSP33. I showed you also that we think that the cells are quite heterogenic in terms of redox status, and that we can actually use redox proteomics to define protein status of cells, and we think that there's collapse in redox status quite early in the age, and maybe this collapse is important for healthy aging. And finally, I would like to thank you and to thank my great group, and especially to Odette, Moore, Mital, and Rosie, whose projects I showed you today, to our collaborators, to funding, and to you. Thank you.